Hi. So I'm in the enviable. I'm Nina. Hopefully I've met. Hi. Yes. Hello. I need no introduction apparently. So, I want to thank the organizers a lot for inviting me to, or allowing me to be here, because this has been an amazing meeting, and I expect it to continue being awesome. And it's basically kind of scary right now, because part of the reason it's amazing is how awesome you've all been, and now it's my turn. I will not be that awesome, and I'm going to beg your indulgence, partially because I threw out my original talk. You'll notice this is not the title I promised you. Based on our conversations yesterday, I thought it would be more interesting for everyone if I talked about this. But it means that I've also thrown my slides together very, very quickly, and there are some things that I'm sure people will want to know, and my answer will be, I would need to ask my postdoc that. I don't know. Also, I need to apologize because my talk was scheduled to end about 30 seconds ago. And it's Friday afternoon, and we are all tired. And so I'm going to try and go quickly. And I'm also going to try and gauge level of interest by feedback. So I'm going to put some of this burden on you guys of active either frowning or smiling or throwing dull objects. Sharp objects, please reserve for if I actually do something terrible. All right, so in that case, without further ado, this is work that's coming out of my lab now, actually. This paper is in review at the moment. I don't have a huge amount of hope that it will get into the incredibly snooty place that we sent it, but then it will hopefully get in somewhere. And so I'm going to talk to you about comparing networks, and I'm going to show you the cases for comparing static networks, but then the generalization to comparing dynamic networks is going to be instant, which is hopefully kind of nice. And I'm going to show you some early results. So these are definitely not meant to be the polished, beautiful, oh my God things. And I'm actually very proud of our acronym. This is N-tangle. And since my postdoc is a physicist, we thought entangled things would be a nice pun. But I was going to tell you the things I'm not going to tell you, but in the interest of time, I'm going to make even that really short. I was going to talk to you guys about a system that my lab's been working on for the last 7 or 8 years, about coming up with a mathematically abstract experimental framework for studying ongoing dynamics for multi-level selection in evolutionary fitness models from networks. And there's a poster about one of those questions downstairs, but we've actually published about 10 papers now on this topic, gradually building it up from the simplest questions to more and more complex questions. And so if you're interested in things like self-organizing networks and ongoing dynamics and the disease processes that live on them, and one of our results is we can show that those are very different from the kinds of processes that live on either dynamic networks that aren't self-organizing but just have an ongoing dynamic, or static networks that are meant to approximate dynamic networks. And I'm shamelessly just going to say, we can show this. And now, if you're a mathematician at heart, we have proofs of why it's happening. But nobody's going to care. So, OK. And we can get metapopulation dynamics out of this. So if you're interested in things like the ecology or evolution of infectious diseases and multiple zoonotic systems and ecosystems, or the emergence of zoonoses, we can do that with different social structures. And it's pretty cool. And you should ask me about it. But not now, because now we're going to talk about comparing networks. So there are some methods already for comparing networks, and some of them are quite good for a variety of things. And we've heard in some of the talks already about some of the ways to talk about them. And the big categories thus far, not the only, but the big categories thus far, have focused on motif-based measures, where what you do is find particular patterns and look for the overrepresentation or underrepresentation of those patterns. A big problem with motifs, especially in the sizes and time frames that we want, is that they get computationally intractable for larger sizes. And graphlets, and I'm shortchanging these guys incredibly. There's a lot that one can say in favor of these, but in the interest of time, I'm just going to be like, things are not great when we use these only. So things are not great when we use only these. Graphlets also, which are induced subgraphs, and you can look at induced subgraphs on subsets of your bigger graph and then compare those, and you get particular types of patterns, and people have done good things with them, and nah. I'm also losing my microphones. OK, so here's N-tangle. And first, I'm going to just describe it very briefly. And then I'm going to start building up what we can do with it, and at the same time try and convince you that it's capturing something meaningful. So what we're going to do is take from our whole graph, we're going to pick a size n. And that size n should be small relative to the whole size of the graph, but it doesn't have to be so small. And then from that, we're going to write down, just for ourselves, all of the possible patterns that we could have on those n, which starts looking like either graphlets or motifs. But then instead, what we're going to do is say, OK, I'm going to ignore directionality, and then I'm going to ignore the pattern. And what I'm actually going to do is just look at the density of this induced subgraph that I get from taking a look at that subsample. And so in the case of things that happen on subgraphs of size 4, I have one thing that could happen with 3 lengths. And now this is all I'm not labeling my nodes. I'm just saying, here's the pattern. And I'm ignoring directionality. So one thing that could happen on 3, 2 things that could happen with 4, 8 that could happen with 5, and 1 that can happen with 6. Yes?
Are these observable events? So you take your network and you see there's only this type of...
So what's happening is whatever network I have, so I haven't specified what my network is yet. Whatever network I have, what I do is I say my sample size is going to be 4. And I pick 4 nodes. And there are different ways to do that that I'm not even going to talk about. But they change the results a little bit, but not qualitatively, just slight quantitative difference. I pick, and then I subsample 4 nodes that I know are connected in some way. So I get rid of things that are totally disconnected. And then I say, what's the edge density for that 4? Then I go back, and I look at my network again, and I draw another 4. And so I'm sampling, and this isn't exhaustive. I'm sampling, which means that it's computationally nice. And then, so this is a visual of looking at what this looks like if you have 3 or 5 or things like that. So then we can do this with some published studies of some people are frowning. Wait, going back, there's frowning. OK, so this is simply if we have an N-tangle size of 4, here's an edge density of 5, because there are 5 edges on that sample of 4 nodes. Whereas a minute ago, if I picked these 4, then I would have an edge density of 3 on these 4 nodes. Better?
Are you sampling 1 node and then getting its neighbors, or are you sampling 4 nodes?
4, but I'm making sure that they're connected. So there exists at least 1 edge. So I'm not caring about anything where the E of n would be 0 on those 4 nodes. Yeah? OK. All right. And then we went to the literature and pulled some really different. So I'm going to disclaim instantly. The animal and social networks that we looked at for this are not generated in the same way, by the same methods, at the same time scales, or even measuring the same things. Some of them are actual behaviors. Some of them are association. Some of them are proximity. Some of them are communication. We just went, what the hell? We're trying to develop a general method. Let's see what we can do with it. So here is a distribution that we get. It happens to be of mountain goats, but it's still in the explaining what I'm doing stage. So forget what the distribution for mountain goats is. What's happening is that what we get is a probability distribution for the edge density on samples of each size. Yes?
When you say probability, what's your universe?
So
the number of links, the number of graphlets with En given.
Yes.
What's the denominator?
So what's happening? So these should be percentages. Crap. Sorry. So this is with an n of 10. This is for 1 n. You're getting this distribution for n equals 10.
What's the denominator? Is it all 4 size 4 samples, or is it all connected size 4 samples?
All connected size 4 samples. Right? So as I sample, what I do is I throw out things where I've picked 4 nodes and they're not all connected to each other somehow. But then the rest of the time I look at this, and I can characterize the not all connected to each other somehow part, but I'm not going to tell you about it now. So I can tell you pairwise if you guys care what to do if you've got really sparse networks, so that this is really difficult to find. But then this is an example for n equals 10. And for n equals 10, we also looked at brush-tailed possums and African buffalo. And there's going to be a slide later about how much I like this one, Asian elephants. So something nice happened here, but we'll get back to that. So, but that's just for n equals 10. That's for one N-tangle size. And what we actually want is to be able to do this for many sizes. So what we get then is a portfolio of distributions for the different subsample sizes. And of course, that's going to tell us something about how sparse the network is. And it's going to tell us something about how connected the network is at different sizes. And it's going to tell us something about the density of those connections. So how cliquish is it? How clustered is it? Of course, and this boils down to clustering coefficient if you choose n equals 3. So N-tangle of cluster of n equals 3 is clustering coefficient, which is already quite nice. And then we have shamelessly stolen these from the literature. My apologies to anyone whose data I've actually taken. How that might be yours, I don't know. I promise that when we're writing this up, we're citing everybody. Some of you have been nice enough to send me data offline. Dave, I think you did that. Thank you very much. And then okay, that was for the biologists. Here's the red deer and here are the whales. For the mathematicians among you, you might have asked, well, is this just a finite size effect because some of these populations are tiny? And the answer is no. Here's some Facebook data, which we did on humans because Facebook data is available and we know what it looks like. And so Facebook data actually does this if you do it by N-tangle analysis. But if you then take the Facebook data and rewire it, which means that for every edge you keep one of the nodes and pick another node at random so that it keeps the same global degree distribution properties and density properties, but then you evaluate the local density properties by N-tangle, you get a very different, and in fact physics kind of predicts this one, a very different portfolio. So this isn't just a finite size, because then you would expect these two patterns to be identical. So still catering to the math people. Is it just the density of the whole network or the size of the whole network that's driving all of these things? Forget whether or not it's finite. What about, well, we've got a network of a certain size. Will all networks of about that size have about that N-tangle? And the answer is no, still doing this with Facebook just because it's a big data set to play with. If I look at all of the N-tangles, and this is a log scale, the N-tangles for size for Facebook, we get a really different pattern from either Facebook rewired, and we have some confidence that the rewired Facebook is about right for a random at that size, because if we look at a random model, in this case I think it's an ER random, but I'm not sure. My wonderful postdoc probably knows that one. You get about the same picture for these, and a really different picture as density increases for that. If you take a look at comparisons with different types of random networks, you actually do get separation in them. So as you try and categorize what does random mean, you can get comparisons for Barabasi Albert. If we take the same Barabasi Albert but then do the same rewiring process we did for Facebook, we get a different signature. Are you?
Sorry, yeah. And just so I understand, in the previous slide, so density, high density is light blue and red low density, is that right?
Yeah, these are the colors are honestly just categories of what we chose to try and highlight that there's grouping. Okay. But yeah, no, they add no particular extra information. They're just there so that it's not all one big block of black. Okay. Thank you. Yeah.
This question is going to bug me as well at the top. How does it compare to graph spectrum? So if you're keeping the spectrum constant, you should get this.
Not quite. So, yeah, she means the spectral image. So it actually doesn't scale. It's not a direct correlate. There are cases where it agrees and cases where it doesn't quite.
Because I mean the triangles, right? It's direct.
Yes. Yes. Triangle. Yes. N equals 3. Absolutely. Absolutely. Yes. If you weren't looking at the whole range of n's that you're going to choose, then yeah, you don't get anything new out of it. But if you start looking at the portfolio of the different sizes of n for the network, then it starts disagreeing as you get different types of structure that would feed into the spectral description.
So is it because you're truncating at connected n-centers?
That's a good question. No, because one of the things that we did was test this on graphs that were only big connected components, and we still get separation, I think, from the spectral answer.
So your samples, right? Because
Yeah.
if you do all
Right, but
n
if
samples, then
yeah.
then you should be getting the spectrum.
So that we're not doing we are not taking all possible groups of, yes.
So the disagreement with the spectrum, is it because you're
No, but I'll show you why after the talk, if you don't mind. Because that does get into, I have to write stuff down and it's math details that some people might not care about. But that's an awesome question. Okay. The N-tangle, or which one? So this is the density. So for the one, so this one's density. That one is the N-tangle. The N-tangle, the Tn that's defined by the soup over all of the E sub n that I defined before as the density on the particular sizes for the n. But they changed from slide to slide, so I don't know which. Sorry. OK. All right. Now, if we believe this part so far, and I am just zooming right through this, and my apologies for that. If we believe this part so far, then it's really quite easy to compare these because what we have are portfolios of curves, and we can just compare those. And so these are curves showing you Kolmogorov-Smirnov statistics, but we checked, and it does not matter what metric you use for these as long as it's a consistent metric. Then you understand what the difference between two curves is, and you're done. Which is lovely because that means that if you've got things that are changing in time, what you then have is the difference between two surfaces. Because what part of the description of the change in time is what is the N-tangle profile going to look like at each point in time that you've sampled. So as you start looking over time, you've simply generated a two-dimensional curve instead of a one-dimensional line or one-dimensional curve, and then you can just take the difference of that. And you can take the difference of that in different ways. So you can either take the entire difference at once, or you can look at the change in the difference between the paired lines sequentially, so that you sort of have a moving window of difference. And because this is well defined, you can just do that. OK, so here are some really, again, these are really preliminary because this is literally just we scrubbed the published literature for networks that had been published and we could look at, and then asked a few people for extra data. I think the mannequins are yours, Steve. So here's one for n equals 8. We had some Asian elephants and some African elephants and some zebras and some mannequins. And this is the N-tangle for n equals 8. And then once we do the N-tangle profile and take the Kolmogorov-Smirnov distance, we can build either a table of what the differences are, or we can do whatever representation you like. And then we can, now here's the important part. We now graph this over all the n of the N-tangles we were summing. And this only went to 20 because some of the networks themselves were quite small. And we want n to be small relative to the total size of the network so that the samples aren't covering a lot of the network at the same time. We actually want that subsample density not to approach the density of the actual network. So here's one of my favorite things about this. This is totally accidental. I have no idea if this is real or not. I kind of hope it is. Notice that both at n equals 10 and n equals 8, Asian and African elephants look really quite similar to each other. My thing I like about that is that if you go to the paper from which we got those networks, it's the same paper, and the conclusion of the authors is that they are strikingly different. The social structures of African and Asian elephants are strikingly different. So we might be getting at something fundamental about how, not necessarily the pattern or the organization pairwise in terms of these dyads, but about what kinds of densities of communication or information or collaboration need to be happening. That's our hope. We don't know if it's true. It's kind of our hope that that's what we're getting. And so these are the actual African, Asian, and then they switch to top and bottom. So African, Asian, African, Asian. And these are the AI thresholds, so association index thresholds. OK, so now, unfortunately, not animal networks, because this is still preliminary for me to be giving this talk to you guys. But we wanted to know how well can we figure out what is generating a network from using N-tangle. And so we didn't just scrub the literature for animal networks. We scrubbed networks. What networks can we get? And the answer is there's a zillion kinds of networks out there we can get. And we feel pretty good about this classification happening. So these are the colors of where we got the network from. And this is a Dendogram representation of, I think this one is minimal spanning tree. Oh, no, hierarchical tree, sorry. Hierarchical tree clustering representation of how close by that Kolmogorov-Smirnov statistic these N-tangle profiles were for these different network types. So we felt actually really pretty good about this system, especially considering that none of these are collected in the same way. None of these are generated by the same things, really. Well, with the difference of Amazon. But that we were really happy about that. Okay, we analyzed. So here's why you might use this. Notice that I haven't told you anything about what being N-tangle similar means. I've said we can find similarity by this way, and I've done that on purpose because what I'm about to try and convince you of is that the definition of a good metric of similarity should be that you can use it to test your own hypothesis of what similar should mean in your system, and that what you can essentially you discover that your hypothesis might be correct by the metric letting you see that similarity as a good cluster. And if it is not a good cluster, then maybe either your hypothesis is not great or the data is not rich enough to let you see it yet, or you're wrong. Just like it could be a bad hypothesis and it could be a wrong hypothesis, and those are possibly not the same thing. But okay, so imagine that we have all of the Facebook networks from university students in 2004 when only university students could be on Facebook, which we do, which is nice. And this is publicly accessible. If anyone wants to play with analyzing this data, it's awesome. But we also know things like the degree of the average network in this, and the average degree. So here it is if we cluster by average degree. And it looks kind of messy. It's terrible. So if we said, well, what happens if we look at the average degree in this network and try and come up with clusters by, and now these colors are the sizes of the universities at which the students were attending when they were on Facebook. So this is not a Facebook metric to which we're trying to correlate. But we might say something about like, oh, big universities promote people who either have lots of friends or small friends versus small universities or something. I don't know. Pick your hypothesis is the answer. If, on the other hand, we asked, can we do this by the network size itself, then suddenly N-tangle orders these perfectly. So this coloring is representative of now the enrollment. Here it was the density. So we had a different hypothesis about what could be driving the similarity. And suddenly we got really good organization and clustering. So we don't just have a randomly true thing where anything you could hypothesize should cluster will cluster well. But when we hit one that seems to work, something really kind of nice happens. And that doesn't just happen for Facebook. Ignore that for a second. We can, and nicely, we have the flip response with archive co-authorship. So here it does seem to be your average degree is a much better a much better idea of similarity than the size of the network, and that actually makes kind of sense as a hypothesis for co-authorship. So types of networks and software calls, so this is which procedures call which other sub-procedures in software, and because programming is a structured environment, you can actually make those kinds of assumptions about this structure. Neither of them is very clean, and that actually is kind of reassuring because neither of them should be because we understand how programming works. So we have something that is a falsifiable hypothesis for how to look at whether or not your hypothesis might be valid. So this is, again, now becomes highly speculative and possibly ridiculous. And I'm going to ask you to bear with me anyway. Here are 37 animal networks. And they're arranged in an order by, I think this might be minimal spanning just closest to Kolmogorov Smirnov. And now we can ask really weird hypotheses, like maybe they clump together really well if you need to have a certain kind of social structure to be a hunter versus a grazer. The answer is no, not good. Don't do that. Apparently that is not true. Maybe it depends on how big your population is. Maybe how big your population is actually, nope, no, that one, terrible. But maybe we could actually see that it actually has to do something or other with the average degree. Not because of the degree distribution, because many of these have similar degree distributions and don't cluster close to each other, and many of them don't. And this is still not great, But we get something closer to bright yellow is up there and dark red is down here, and then it gets kind of mushy in here. So maybe we're getting at something. Maybe we should start to investigate that. Maybe the idea of similarity should be that it lets us go, that is an intriguing hypothesis. That seems to have popped out. I wonder what did that, in the same way that we currently use clustering algorithms, like k-means, to do that. So maybe we can do it this way. And when we look at normalized, yes?
Sorry, quick clarification.
Yeah.
This is just the one given small n.
Yes, sorry. But you can do this, I'm showing that as the example, but you can do this over the whole panel. And in fact, in a second, I'm actually going to switch. Oh, wait, actually, I'm sorry. No, by now this is looking at the Kolmogorov-Smirnov distance over all n. Over all little n for one big N for each population, but that's different for the different populations.
Yeah, but you truncated some little n.
Yes, which we did as a percentage of the total size of big N. So it's a different little n cap for each one, but then the Kolmogorov-Smirnov distance is normalized. So you can still compare, which is nice because that means you can also compare things over different sizes, right? The animal networks to the size of the internet, really different, and yet I could show you that one graph of how far away by Kolmogorov-Smirnov distance is archive collaborations from elephants, right? So we don't need the same size, we don't need the same generating mechanism, but we also don't know what our question is. And so suddenly now when we come to normalized degree, where now the size of the underlying population matters, now we get something that I really do think is quite a nice ordering of yellow with a little bit of orange but not much, orange with a little bit of yellow but not much, red with a little bit of orange but not that much. So this gives us some more faith that there's something structural. Notice that normalized average degree is a structural property, so it's not that surprising that we're going to get that out of looking at a density measure. But we got one, which is nice, because these are 37 complete, well, most of them, so at most 2 of them, are gathered on the same data in the same way, which means that this is really difficult data to deal with. So now it can help us also pick an organizing principle for size. So if you want to ask, what is the important collaborative unit for your population, or what is the important operative, not distance away in the network, but distance away in terms of the average subsample for things to organize well by that hypothesis, you can compare. So now is the graph with just the one n size. For n equals 5, we get kind of messy things. For n equals 10, it's starting to look a little bit better. For n equals 12, it's looking really quite good. And for n equals 15, it's starting to look messy again. So we clearly sort of we went past it. So the answer is, for these networks, and that's because some of these are in fact quite small, by the time we get up to n equals 15, nothing really interesting is being told to us about this hypothesis, just this hypothesis. Other hypotheses might be really interesting here for n equals 5 or n equals 15. But again, if we try and do this clustering, it turns out it's never a good idea to say that your population should organize in a particular way just because you're hunting. As opposed to grazing. All right. You can do Dendogram kind of things with this if you like that presentation better. I actually find this less natural, but ta-da. OK, so, this one. We decided to ask an ecologist what he thought of this. And so we asked Peter Morin, who is amazing. I don't know if any of you know him. He's awesome. I recommend him for everything in life. But he also put up with me rambling at him about this idea, and he went, okay, I guess that's kind of interesting, but where's the ecology for your hypotheses? We had structural hypotheses, and he went, where's the ecology? And I went, great, what are some good ecological clusters that we might want to hypothesize to get out of this that aren't as stupid as hunter-grazer kind of classifications? And so he came up with 4 with us that are. Here's the other bad thing about this. When we came up with them, we then did a back of the napkin, ask Peter Morin where he thought they fit calculation. So these are clumped. Each one has red is high, green is intermediate, and blue is low. For how many competitors do they have in their ecological niche? Keep in mind, our metric for this was Peter Morin thinks this. Not even asserts it. Peter Morin is willing to say, go try this. Turns out, no, we really don't get very good clustering by amount of competition in your niche. Your density in the landscape Also not great. What you eat. So are you a carnivore or herbivore? That gets back close to hunter-gatherer, but not quite. Where are you in your trophic level? Are you an herbivore? Are you a carnivore? Are you an omnivore? None of these really work that well, but hey, we tried them, and now we know none of these really work that well. And then we did a really naive linear combination. We said, what happens if it's actually kind of important to take some of each of these into account? And we know that it's not actually going to be linear, but we would have had to waste much more of Peter Morin's time if we actually tried to figure out how this might affect social structure. But suddenly we can get something that isn't that bad. And so here the coloring is by what? Here the coloring, I think, is by overall. No, it can't be by overall N-tangle. Oh, it's the normalized degree. That's what it is. The normalized average degree is the coloring on the left, and the linear combination of the random Peter Morin is wise variables are on the right. And we get not great clustering, but still something better than each of the independent ones, where we go, oh, look, a chunk of red, and another chunk of basically red. And if we look at the species inside those chunks, there are things that seem somewhat logical about some of them, and then ones that seem not at all logical, like why guppies and giraffes are really closely related. I have no idea. But they're probably different association metrics, is my guess. So what do we have? We have, hopefully, a proposal for a scalable metric of similarity among networks of different sizes and types that can be easily generalized over time. And it's computationally tractable because what we're doing is sampling. It's not just a correlate of the other known metrics we've checked. And if anyone cares, we now have literally like 20 pages of what will eventually be appendices of some god-awful paper of we checked this, and it's not just that. And if you know this information already from normal metrics, you haven't accidentally gotten this. And but it can be used to correlate with hypothesized biologically relevant characteristics for why you might have this kind of social structure. And at what size and at what unit of analysis might each one of those be important? And then what kind of function for looking at their combination might get you the best clustering by doing this is a way to start thinking about hypothesis testing for both static and dynamic comparisons of these things. And then the people circled in yellow are the wonderful people who did the work for the poster downstairs and that I was going to talk about the talk today. And Lazarus Galos, who is amazing, is my physics postdoc, and he did the work I actually talked about with me today. And then there are other people in my lab who are awesome, and thank you so much.